And if you've had A&P, like that, this is something that you've heard already. But again, let's just go back and think about that size thing again. So we talked about this at the beginning of 10. So we said smallest molecules, simple carbs. The sugars. Far and away, the smallest of the molecules that we've been talking about that are part of your nutrient molecule group. Then from there, we said lipids. The fats and the oils. They're bigger, 5 times bigger. Then proteins come into play. It's hard to give just a single example of protein because you think of like meat, I guess. Typically 2 to 5 times bigger than lipids in terms of molecule size. From here, the only other big one we talked about, the one I said is huge, is what? It's not the simple carbohydrates, it's those complex, those polysaccharides, the starches, the fibers, huge molecules compared to these. One of the nucleic acids, RNA, is a little smaller than the complex carbohydrates. RNA found in the cell, not really so much of a nutrient molecule, but one that you do see as a molecule in the cell. This is the molecule that's used to make proteins. This is the short part of the genetic information that's used to make a single protein. Then you get to the complex carbohydrates. Just remember we said that those are like thousands of sugars. Always made of glucose. It's like 1,000 glucoses in a molecule. So RNA is a little smaller than that. But far and above is DNA, which is deoxyribonucleic acid, which is where the DNA comes from. Size-wise, this is on the order of 5 to 10 times bigger than a complex carbohydrate. So these are really huge molecules, even larger than the complex starches that we've talked about so far. So this chapter introduces the structures of these two and then how they're used and why your cell, every single nucleated cell in the body, has both DNA and RNA. So whether you're talking about DNA or RNA, they call them as a generic group, they're called nucleic acids. Just like proteins have building blocks, which are your amino acids, nucleic acids have building blocks, those are called nucleotides. Nucleotides are made of a phosphate group. So remember that the phosphate group is a PO4 2-. That is a polyatomic ion. It's 3-. It's got some kind of sugar. It's a 5-carbon sugar, so it's a pentose. We actually looked at these ones. It's either ribose or deoxyribose. So we talked about those way back in the carbohydrate chapter. So you've got a phosphate group, a sugar, and then it's connected to what's called a base. Now, when people say base, they always think, well, what is it, like sodium hydroxide or potassium hydroxide? It's not like acid and base. It's an organic base. So it is a molecule made mostly of carbon. And there's there's really 5 different potential groups. They are adenine, thymine, cytosine, guanine, and uracil. Oh. So there's the list that's there. So these are just the 2 categories of them. So you have adenine. Notice all those nitrogens. 5 nitrogens in an adenine molecule. Remember that all the little corners, every end, every corner, those are all carbons because it's just showing the skeletal structure. Guanine, lots of nitrogens there too. Double ring structure. Cytosine has just a single ring along with thymine and then uracil. These 3 you find in both DNA and RNA. So adenine, guanine, and cytosine are found in both of our nucleic acid groups. These are the 2 that are a one or the other. So DNA only has thymine, RNA only has uracil. So you have T's in DNA, you have U's in RNA. They call these nitrogenous bases. They're just organic molecules that have a lot of nitrogen on them. So again, here, there's carbon, there's nitrogen, there's oxygen, and there's hydrogen. So you have the CHN, you have the 4 of the main elements like you see with proteins. So when you put these together, well, there's the sugars. Remember we looked at the ribose and deoxyribose. The only difference between ribose and deoxyribose, ribose has an alcohol group on carbon 2. Deoxyribose does not. So its hydrogen has been deoxied on carbon number 2. That's really the only difference. Remember that numbering pattern. So when these get linked together, you have a phosphate linked to a sugar linked to the base. So here's your phosphate connected to the sugar, connected to a base. So phosphate and sugar, and off of the sugar is always the base. This connection, this linking, forming these types of bonds, just like with proteins, condensation reaction. So condensation reactions form what's always a product in a condensation reaction? Water. So remember condensation, like think of water forming. And so in this one, they actually color the water. So you see the alcohol group on the sugar, or the hydrogen off of the sugar, the alcohol group off of the phosphate. Those get pulled in order to form that link. So I kind of think of them like that. So you have a sugar linked to a phosphate, and that phosphate's always linked to the base. So that's a nucleotide. That's a building block. But now, nucleotides get linked together. So a sugar gets linked to a phosphate below, which is linked to a sugar which has a base. So that's 2 nucleotides. Another one gets added. So just like where we had amino acid 1 linked to amino acid 2, linked to amino acid 3, and forming this chain, you can form a chain between sugars and phosphates, and that base is always kind of the one that sticks off. So the base isn't part of the chain, the base is sort of like this little extension always off of the sugar. So you can see in this one, they show how they link them together. So in this one, the top one, phosphate sugar in the base. Next one down, phosphate sugar in the base. And so it is the 5' or the 3 carbon that connects to the phosphate. That phosphate is connected to the 5th carbon of the next of its sugar. And then if you have another one that occurs, you'll have another phosphate connected. So you'll always have them kind of connecting this way. So you would have up here, you might have up above, you may have a sugar up here and a phosphate off to the side and a sugar and a phosphate. But always off of the sugar is where the base extends. So there's one. There's the second one. So what you end up seeing is this. I'll start off with a phosphate to a sugar, to a phosphate, to a sugar, to a phosphate, to a sugar, to a phosphate, to a sugar. And always extending off of the sugar all the way down the chain, you'll have some type of base that sticks off. It might be an adenine, it might be a thymine, it might be a guanine, it might be a cytosine, but it's always one of those organic bases. This is considered the backbone. That's considered the chain. And the base is what makes the sequence of the nucleic acid. So you look in this one, so you can see, here's your backbone. Just like we had with the amino acids, that nitrogen, carbon, carbon, nitrogen, carbon, carbon, that's kind of what formed the backbone. Well, here, the sugars in the phosphate form the backbone, and then you have the bases that stick off. They even shorten this. So they'll shorten it down so that it's just little SP, 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 kind of the way that I draw it, and then just the base sticking down. Or they can even shorten it further and not even show you the backbone. So in that, all they're showing you is just the order of the bases. But just remember that that's not all that it is, that it's all those bases are actually sticking or hanging off of that sugar phosphate chain. It's just a way to simplify. When they talk about these ends, that is the carbon group that's extending up. So from that sugar, that's the 5th carbon that extends up. And then at the very end, sticking down off of the phosphate, which would stick to another sugar, that would be the 3' end. So all that's referring to is like where you began to connect them. They always get connected from 5' to 3'. And so they say that you can kind of like read from one side to the other. So there's a direction. Very much like with proteins, you have NCC, 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 nitrogen, carbon, carbon, and then you have the R groups hanging off. So there's like a nitrogen side and there's a carbon side. So there's a beginning and an end to a protein. There's a beginning and an end to your nucleic acids as well. So you can't take it and like flip it backwards. So it would actually be oriented, the sugars and phosphates would be oriented slightly different if you flipped them around. So things that they started to study, and in fact, nucleic acids are some of the newest in terms of the understanding. Some of the most recent discoveries about the biomolecules, molecules found within cells, came in the 1900s. So this isn't something, so remember like Marie Curie, they were right around the turn of the century, about 1910. So this didn't start work, they didn't start doing things until the 1940s, 1950s, and it was really late 1950s, 1960s before the double helix was actually even introduced. So one guy that was working on these took DNA, because they knew that there was like this genetic material. So they had isolated DNA, and he started looking at what it was made up of. So they determined there was phosphates and that there were sugars. He also noticed that they had these organic nitrogen-containing bases, and he found that if he separated and measured the amount of adenine versus the amount of thymine, they were always the same. And if he measured the amount of cytosine and the amount of guanine, that they were always the same. So it's like, well, there has to be some kind of balance between to keep them always in this, whether we are talking about human DNA, if we're talking about frog DNA, if we're talking about chicken DNA, you're always seeing adenine and thymine have the same amount, cytosine and guanine have the same relative amount. So in the 1950s, two men by the name of Watson and Crick, they didn't actually do a whole lot of experiments on this. In fact, they went to a lot of meetings. They hung out, and they were both PhDs. They were scientists. They just actually didn't do a lot of the research on this. Instead, they were really good at taking information and pooling it together. So they had somebody that was actually doing research trying to create crystals of DNA and look at it under special microscopes to see how it was positioned. Then you had Chargraph, who had published about the adenine and the thymine, cytosine equal guanine. And so people were trying to figure out how it would fit, because they knew if they took if they thought, well, if it's two strands and they put those two strands, the adenine and the thymine don't match up. Their structure doesn't allow for them to interact. Same thing with cytosine and guanine. What these two came up with is they said, what if you took the two strands and you flipped them? So that there's actually, this strand runs from 5' to 3', and this strand runs from 5' to 3', and they run the opposite direction. And if you do that, then suddenly these base pairs line up exactly the way that they should. And so if you look at this top picture, so the top picture shows you how adenine and thymine, not if they're this way, but if you take the strands and flip them this way, adenine and thymine line up and they hydrogen bond. So you see that the adenine has a nitrogen with a hydrogen, and then right across is a double bonded oxygen. So those two can share that hydrogen and create a hydrogen bond. Same thing with that second pair, the nitrogen on the thymine, hydrogen bonds with the nitrogen over on the adenine, and that's going to create an attraction. Adenine and thymine create two hydrogen bonds in between, and if you follow down, you see how at the top they have 5' up here, 3' up here, which means that those strands run in the opposite direction, but it lines them up perfectly so that you get hydrogen bonding, all along with those bases. So the purple is the sugar and phosphates. So you have sugar, phosphate, sugar, phosphate, sugar, phosphate, and sugar, phosphate, all the way down. But off of each sugar, you have the base that extends across and hydrogen bonds with the base on the opposite strand. Here's how cytosine and guanine look. So they actually form 3 pairs of hydrogen bonds. And you remember we said hydrogen bonds aren't very strong, but when you have a lot of them, that's what holds up the water bug. That's what creates raindrops. That's what creates that ability to water ski and skip a rock, hydrogen bonding. So if you only had a few, it doesn't make as much of a difference. But look at this little span. See all the little blue dot connections. So all of those create this relatively strong double strand. In fact, the strand then begins to twist, so they called it the double helix, because it has this twisted arrangement that is further stabilized by that hydrogen bonding. So Watson and Crick is pretty genius that they could put all of that information together, considering they really didn't do things. But they did. They went to meetings. And they heard people publishing, oh, I did this, and I did that, and I did this. And they were like, So they put it all together and then eventually published, and what they said turned out right. And some people were a little mad because they didn't do very much of the research. But it really then allowed this big step forward in this understanding. The thing to understand is think one nucleotide of DNA is one pair, sugar pair, phosphate pair, and pair of bases. So they call them base pairs. So the AT, that's a base pair. GC, that's a base pair. Human DNA, you have 3 billion base pairs in one nucleus of a cell. And you originate from one single fertilized egg, and that's where that DNA is arranged. And so when that cell divides, each new daughter cell gets 3 billion. So those cells, before they divide, they have to replicate their DNA. So you've got to take 3 billion to make 6 billion. And that way when they divide, each of them get 3. But 3 billion. 3 billion of these bases. So in this one, what we're looking at, we're really only looking at what, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, like maybe 16. So it's a pretty short strand in comparison. So you would have to take this and have 3 billion. So there's not one long strand. It is arranged as chromosomes. It's arranged on 23 pairs of strands. That's what chromosomes are. So this organization, what they call the primary sequence or primary structure, just like with proteins, there actually is a primary structure to DNA. And the primary structure is just the order of the base pairs along the nucleotides. Anytime you see A, every time you see an A, on the opposite side, you would see a T. Anytime you would see a C, on the opposite side, you'd see G. So you can just make a strand. Just randomly adding them in. Whatever though you find on the opposite side, it'll always be the complement base to the one that's on the left. So A will have T, T would have A, C would have G, G would have C, G would have C, A would have T, T would have A. And between these bases are hydrogen bonds. So those hydrogen bonds are what help to hold this together. A and T have 2 sets of hydrogen bonds. G and C have 3 sets of hydrogen bonds. And so hydrogen bonds are not very strong, but when you have 10, 10 is the equivalent of a covalent bond. So in that, we have 5, 10, 15, we have 20. So it's like having 2 covalent bonds holding this area together. So I almost think of them as kind of like magnets. So they kind of go boop, boop, boop. And you can actually pull them apart, but they're going to like wind themselves back up. Unless you physically pull those strands apart, they're more stable hydrogen bonding than they are as single strands. So that's the primary. So you would say the primary structure that's ATGC, ATTACG. You would just like read the pairs. That's the primary sequence. Secondary, though, is that this helix doesn't just look like a ladder, it twists. So the twist is the secondary structure. The twist of the helix. And remember again, if it was straight, it wouldn't have much give. But having this twist, now, kind of like the coil, it has a little more stretch. You can take this helix and pull it a little bit, and it can go back. You actually do have, kind of like with alpha helixes and proteins, there is some hydrogen bonding between the backbone to keep this. So it's going to give a little more flexibility to the molecule than it would look if it was just like a straight ladder. I just can't draw it as a double-stranded helix because I don't have that artistic ability. But like I said, you have 3 billion of these, 3 billion base pairs. If you had it all in one long strand, it would extend about 6 feet. And that's from one nucleus. So you would have a strand that would be this long, and it would be so incredibly thin, so it would get knotted and torn. It could get damaged easily. So having it in the coil helps, but then it gets organized within the nucleus of the cell, and it greater by being wrapped around histones. So there are these small, and you can kind of see them in this. So these little yellow, look like little like marbles. They're actually proteins. The histones are small, positively charged proteins that the DNA wraps around. And what it does is it takes this really long strand and it helps to condense it. It helps to shorten it. It helps to organize it. So the odds of it twisting and tearing go down. So this wraps the DNA into a structure that's called chromatin. So chromatin in the cell is this loosely packed DNA it's wrapped around these histones because that's going to help to keep it from getting twisted, getting knotted, and torn. Supercoiling occurs as a way to further organize the DNA when it has to go through mitosis. So when one cell splits to two, you can't have 3 feet worth of strands. Getting those organized would be really difficult. So it takes this wrapped DNA and it coils it back on itself. So it sort of coils the coil. And when you get more and more coiling, that actually causes the strand to supercoil. And I didn't bring my rope with me, but I have like a string. So like, and you can try this. Like if you take just a regular piece of string, and if you just start twisting one end, you'll notice that it kind of like starts twisting up and twisting up. If you keep twisting and keep twisting, it starts to get smaller and smaller and smaller, twisting back on itself. That's actually what ends up happening to the DNA molecule. And it coils up so much that instead of it being that 6-foot-long strand, it is microscopically thin. So it's thickened and it becomes visible when you stain it in the structure of chromosomes. So this really just helps to organize DNA into thick, short, pieces called chromosomes that can be moved around. The only time you see chromosomes is during cell division. It only happens during mitosis. So it only happens when the cell has to tightly wrap its DNA in order to be able to divide. That's considered the tertiary structure. The highly folded structure of DNA wrapped around those histones, keeping it really organized within the nucleus. So different animals have different numbers of chromosomes. Humans have 23 pairs. And so that means that it takes that 6-foot piece and it's split into 23 pairs. So it's not really 6 feet long. One single chromosome, you actually did this, or you're going to do this in lab tonight. You did like a math problem to figure out about how long a single strand is. And they're about 2 inches long, 2 to 3 inches long is the average strand of DNA found in a chromosome. So it's not really thick, like really long. But when you're talking about a microscopic cell, that's still an awful long piece to have packed inside. So organizing it helps to keep that from being twisted, torn, or getting knotted. So we really talked about DNA. So let's do a comparison of RNA. So we said that DNA is a double-stranded helix. Well, RNA is just a single strand. DNA has adenine thymine, cytosine, and guanine. So its bases are A, C, G, and T. RNA, though, its bases are A, C, G, and U. DNA sugar is deoxyribose. The sugar on RNA is ribose. They're linked together the same, sugar phosphate with the base hanging off of the sugar. It's just the DNA has those two strands that run in opposite direction. RNA is just a single strand. In the cell, DNA stays in the nucleus. So the nucleus is sort of the core of the cell. RNA, on the other hand, it's made in the nucleus, but then it travels to the cytoplasm. So it doesn't stay in the nucleus for its entire span. Size-wise, so DNA, we said 3 billion nucleotides, 3 billion base pairs in one cell. RNA is much smaller. RNA is just a short sequence anywhere from 100 to about 450 nucleotides. So much smaller in size than DNA is. DNA is always present. From the time that the cell is first made until the cell divides, it's always been there. Okay, so it's kind of like this constant thing in the nucleus. Ooh. RNA, on the other hand, it's made when it's needed. It's made from the DNA, and only when it's needed. And if it's not needed, the cell will break it down. So it's only present whenever it's needed. And this leads to the function. DNA contains all of the instructions to make everything that the cell needs to have. It is the genetic information. DNA controls things like cell division. DNA controls protein synthesis. The development of the cell, controlled by the DNA. Everything that is needed to get you from a single fertilized egg to the trillions of cells that you are today is all from the DNA. But RNA has really got just a specific purpose. It's made by the DNA, or made from, and then it can be used to make a protein. And this process then means that there's like a flow of genetic information. So DNA is used to make RNA, and then RNA can be used to build a protein. And so by proteins, we're talking about enzymes, antibodies, collagen, hemoglobin, insulin, all those proteins types and all those examples that we were talking about in the last chapter, for the cell to be able to build them, they start with the DNA. The DNA contains regions that are called genes. Genes are sequences that have the information that is necessary to make RNA, which then can be used to build a protein. They call this whole process protein synthesis. So we'll talk about, why is it keep jumping? Talk about protein synthesis, how proteins are made, and then we're going to talk about mutations. What happens if there's something wrong with the DNA? What happens if there's a change? What kinds of things can cause a change in the DNA sequence? And what effects can that end up having? So that's kind of like the last part of chapter 11. Last time we stopped, we had talked about DNA and kind of doing like a comparison of DNA and RNA. DNA being that double-stranded helix, we talked about like the helix shape, how it forms chromosomes, whereas RNA is just a single strand. DNA is located in the nucleus, and there it remains. The nucleus of every cell, it has its own enclosure, the nuclear envelope or the nuclear membrane. RNA, on the other hand, is actually made in the nucleus. It is made from DNA. However, it is transported into the larger compartment of the cell, which is the cytoplasm. So that's sort of just the interior compartment. Remember other things about DNA. We have DNA as deoxyribose. RNA, on the other hand, is ribose as the sugar. DNA has that ACTG. Thymine is one of the organic bases. RNA has uracil. So it's another variation or difference. Well, 3. A newly formed cell has 3 billion nucleotides. And in humans, it's on 23 pairs of strands. Different animals have different numbers of strands, but humans have 23 pairs. RNA, on the other hand, is much smaller. So RNA is made from DNA, but RNA is just sort of a short form. It is only going to be made from a short sequence of that DNA, typically RNA strands being single-stranded, and you're really talking about somewhere in the neighborhood of 500 to about 3,000 nucleotides. Much, much smaller. Made when needed. This is because DNA is the complete set of genetic information that is needed to make all proteins that any body cell might need. RNA, on the other hand, is just the amount of information needed to make one protein. So it's really just a short version of genetic information used to make just one. So in sort of the flow of things, DNA is converted to this short version, which is RNA, and then that molecule is what is sort of the code or the instructions in order to make a protein. So remember in the last chapter we were talking about proteins like collagen and keratin and elastin and hemoglobin and all the different enzymes that you need. The RNA, you have a sequence of RNA that is used to make one of those, and that comes from the DNA. So the sequences that are used in DNA are called genes. So a single gene is used to make a strand of RNA, and that's the information to make a specific protein. So I've got a handout to kind of help you organize it. So at the top, sort of this flow. So in this flow, the first step going from DNA to RNA, this step, it's really going from one nucleic acid to another. Not even knowing why. Okay. So in this step, this first part is called transcription. So transcription, think of it as taking one type of nucleic acid and converting it to another. There is a special enzyme that's involved in this process. This enzyme is called RNA polymerase. Really? So RNA polymerase, from its name, it actually tells you what it does. So the ASE on the end of RNA polymerase tells you that it's an enzyme. And so polymer, when you think of poly means what? Many. So this is an enzyme that makes a polymer of RNA. So its name is actually telling you what it's doing. It's making a polymer of RNA. I'm not sure what it's doing. It's working just fine. Just can't today. And it switched back and forth last night just fine. Not an issue. There. Nobody knows why. All right, so this is transcription. So if DNA is always in the nucleus of the cell, this is where transcription happens. And the enzyme that's involved is an enzyme that's called RNA polymerase. So it's this step. So what happens is there's a sequence, and there's lots of them. You have somewhere between 20,000 and 25,000 genes in the DNA. So one segment of this, when you need to make a specific protein, one segment, this RNA polymerase unwinds the DNA. And it does this at the beginning of a gene. So it unwinds the DNA. It uses one of the strands. It doesn't use both. So that DNA, remember, is the double-stranded helix, and it's wound up around those histones. So what this enzyme can do is it unwinds and it pulls the two strands apart. This enzyme then uses one strand of the DNA as a pattern to make a complementary strand of RNA. And by complementary, they're going to be able to hydrogen bond. So the matching occurs because the bases can hydrogen bond as this new strand is being formed. So if the DNA has a T, the RNA polymerase will match what complementary base? An A. Because A and T can hydrogen bond. The shape of the two molecules, those bases, remember in the double-stranded helix, you can have the hydrogen bonding. Then the enzyme slides down. If the next one's a G, what will the RNA be? So this is like considered the coding strand is the DNA. So if there is a G on the DNA, then the RNA polymerase will match the complementary base, which is a C. If the next one's a C, you'll find a G gets paired off with it. What if there's an A? Then you'll have a U. So if the DNA is an A, then that will match a U. And it just continues down like this. So there it starts at one point and it just matches for every base that's on that DNA coding strand. You match an RNA base to it, and you begin to link them together so the enzyme links it and forms an entire strand, sugar, phosphate, sugar, phosphate. Off of each sugar is the correct nucleotide. Once it's finished, and there's actually like sequences on the DNA that indicate this is the starting area, there are sequences on the DNA that indicate this is the end of the gene. So the RNA polymerase knows where to start, and it slides along, and then it knows when to stop. So when it stops, you'll have this sequence of RNA that is like attached to the DNA. But when the RNA polymerase separates, it pulls the RNA off, and those two DNA strands then can wind back up. So they end up going back into their double helix strand arrangement, and this piece of RNA, so at the end, the enzyme releases the RNA, and the DNA winds back. This strand of RNA that's now this newly made strand from that section of DNA, that strand of RNA, this is called messenger RNA. Short term, they call it mRNA. And you can think of it, it's carrying the message of the gene. So it's carrying the message of the DNA strand, and that's going to be the strand that's used to make your protein. But it can't stay in the nucleus. It leaves the nucleus and it goes into the cytoplasm. So the cytoplasm is sort of the larger compartment, the interior of the cell. And the reason for this is because this is where the organelle is located that actually makes the protein in the second step. So it's called transcription. It's where this RNA polymerase takes a piece of DNA and uses it to synthesize a complementary strand of RNA. That complementary strand of RNA is like a short piece compared to the whole entire DNA. And that strand now is moved out into the cytoplasm so that we can use it for the second step. So here's a DNA gene. And we're just going to say, so you know that this strand is way longer than this. So there's like, probably 10 million or so on this strand. But we're just looking at this one little piece. So this piece is where our gene is located. So we're going to use the bottom strand as the coding strand. So this strand is the coding strand. So if you were RNA polymerase, what would be the first base that you would match? So you'd have A, then U, then, and so just go along. So there's a T, we'll put an A. There's an A, we'll put a U and we'll link them. Then there's a C, so we'll have a G and it'll be linked together. Just continue all the way down to the very end. Use the bottom strand as the coding strand. Okay, so everybody went through and did the list. And that's all you're really doing is just matching from that coding strand. You're just making a complementary piece. There's a beginning, there's an end. So the RNA polymerase releases this new mRNA and it goes out into the cytoplasm. So here we're going to do the second step. And so the second step is when you're going from RNA to proteins. That second step, RNA is a nucleic acid, proteins are amino acids. So we're actually going to be converting it to a completely different kind of molecule, a different type of language if you want it. That is called translation. So I always think of like you translate from English to Spanish, and so it's like a different language you're translating it into. This is translating it from a nucleic acid into a protein. Now this is going to take place in the cytoplasm. So this takes place in the larger area of the cell. And instead of just a single enzyme, there's actually a couple of things that have to come together and play in to do this. The major organelle, it's the smallest organelle in the cell. They look like little specks. Does anybody know what the name of that organelle is? You can have millions of them in one cell, especially if the cell's really active. They're called ribosomes. So ribosomes but we also need this mRNA because this is telling us the pattern. And there's a third molecule that comes into play. They're the ones that are responsible for carrying the amino acids to the ribosome. They're called tRNA. So here we need to look back at the PowerPoint just to kind of define these three things that are used in translation. The first one is this ribosome. So a ribosome is actually a mixture of RNA and protein. So these two things together form this like kind of double subunit molecule. And I'm not going to lie, it looks like a hamburger bun to me. Like that's just the way that I always think about it. You've got like the large top and then the flatter looking little bottom. And what the ribosome does is it actually attaches to that mRNA strand. And so it literally, like the small and large, they just sort of like slide over top and underneath that RNA strand and kind of sandwich it between them. So these are the two. So it's made of a type of RNA that they call ribosomal RNA. That's also made from DNA. It's made from a specific part of the DNA. And you just churn out lots of this, but instead of it making protein, it actually becomes part of this organelle, the ribosome. So the ribosome is really kind of like an enzyme because the ribosome is going to interact with the mRNA, but it's also going to allow for protein synthesis to occur, and it is the reason that peptide bonds form. So ribosomes, they're enzymes, and they're going to help to form the links between amino acids. Those are the peptide bonds. So when you think of a protein, amino acid 1, link to 2, link to 3, link to 4, those links are the peptide bonds, and it's the ribosome that forms them. So we have mRNA. We also have this RNA called rRNA, which just stands for ribosomal RNA. But rRNA is strictly found just in ribosomes. But we've got another one. We've got a third kind of RNA. Even though RNA is really pretty small, there's really 3 kind of different types that all work together to make this protein. The last one I told you is tRNA. So T stands for transfer, M stands for messenger, R stands for ribosomal, T stands for transfer RNA, and the job of a tRNA is to transfer amino acids. They're kind of like the carriers. They are going to bring the amino acids to the ribosome so that the ribosome can link them together in the correct order. So the way that this looks, a tRNA molecule, that looks like way more complicated than I can draw it. So whenever I draw them, because it's kind of got the molecule itself, tRNA always has an amino acid attached. And then it also has this region. This is the region that can interact with the mRNA. So this region has to be complementary to the strand on the mRNA. So you notice in this one, like U and A, they hydrogen bond. G and C, they can hydrogen bond. So they fit together. And it is actually a 3-base sequence on the mRNA must be complementary to the tRNA sequence. They call those areas codons. Okay, so that 3-base sequence is called a codon. This group here. And it's always in sets of 3. So it's not just one base has to match, because then you would have a 1 in 4 chance, because it could have been in either an AU or a GC pair. But instead you have a 3-base set that has to match in order for the tRNA to interact with that mRNA. The sequence determines which of the 20 amino acids the tRNA carries. Now, the way that I always draw them is I kind of draw it looking sort of like this, with an amino acid on the end, just because I can make it look kind of like a hanger type of structure. So it kind of has that structure, sort of, kind of. It's just easier to draw than their little wiggly lines going all over different places. So this entire sequence is an RNA sequence. This entire sequence is, again, made from DNA, but instead of actually being used to build the protein, it's used to form these carrier structures, and their job is to carry the amino acid to the ribosome. So that's why the term transfer RNA, just think of it as it's responsible for transferring amino acids to build a protein chain. So we've got those three different structures. So in translation, what we're going to have happen, let's go back to our, here. And we'll first look at this green sequence that I've got. Okay, so that's the mRNA. So there's 3 steps that happen in translation. There is the beginning, the middle, and the end. They call it the initiation, the elongation, and then termination. The beginning of the protein synthesis step is when the ribosome binds to the beginning of the mRNA. And I specifically wrote this structure because the ribosome always binds to a sequence AUG. So AUG is always the beginning sequence on any mRNA strand. So AUG is kind of like start here. So that's found consistently. So the ribosome, here's the big top and the little bottom. So the ribosome will attach at that point. The large subunit and the small subunit come together to sort of sandwich the beginning of the mRNA, and it's always at that part. So that sequence will allow a tRNA to interact only if it's complementary to that AUG. So if we have our tRNA strand, it's carrying an amino acid, what would its sequence need to be complementary? A would have a what? Nope, can't have a T. This is RNA, so it would have to be a U. So we'd have a U, and then the next one would be an A, and the next one would be a C. So UAC is what would be on the tRNA, and that would allow hydrogen bonding, and that tRNA can fit. If it was like UAA, it would kind of be like this. It wouldn't be able to sit down flush, and if it can't sit down exactly even and match all 3 of those, the ribosome just kicks it out. So if it doesn't match, it's not able to remain in place. So this one, the only tRNA that'll fit there is one that matches or complements that AUG. So they actually determined which structure these accounted for. Come on, there. They call this the genetic code. So this looks at all the possible 3 base sequences and what amino acid the tRNA would be carrying. So if you look at this, so there's the first letter, the second letter, and the third letter is how they came up with the chart. There's a total of 64 possible combinations. But if you find AUG, what do you see that it says? So see here, oh no. So see the AUG? So AUG is like the start sequence. So AUG is going to have a tRNA that's carrying an amino acid that has the abbreviation MET. So that stands for methionine. Methionine is one of the sulfur-containing amino acids. So this is going to be MET. So there's a methionine. It's always the first amino acid because it always matches that AUG. Once this happens, we said that this would be a U, a C would be the complement. Once we have one tRNA that sits, the ribosome will slide along. And it always moves 3 bases. So the next set of 3 bases is AUC. So if you just take and slide the ribosome down, the first tRNA is still there with its methionine. The second one's AUC. So look back at your code. AUC, what's the amino acid that AUC would be carrying? What's the abbreviation for it? So CILE. So CILE right here, couple up from the other one, that stands for isoleucine. There's 20 different amino acids. Each one has its little 3-letter abbreviation. So that's going to be the one that sits down in the second spot. So you'll have a ribosome, the tRNA comes along and it's carrying ILE attached to it. So what is the tRNA's code? If it's AUC, A would be a U, U would be A, and C would be G. So you see that those are different. So that tRNA would have to have a sequence that's complementary to the mRNA strand. So when two amino acids are side by side on the ribosome, so you have the ribosome, the two tRNAs, and you've got these two amino acids side by side, this is when the ribosome acts like an enzyme. So the ribosome acts like an enzyme and forms the peptide bond. So it does this to link these two amino acids together. And in doing so, it breaks the connection that the first tRNA has with its amino acid. So it actually breaks this connection off. So now ILE is connected to MET, but that left tRNA is like, it's called empty because it's like had its amino acid removed. So this one just ends up falling off. So this starts the process of elongation. So once you form a new peptide bond, the ribosome slides down and the empty tRNA kind of falls off the back. So if it doesn't have an amino acid attached, it's not going to stay put. It just ends up dropping off. It can actually have a new amino acid put back on it, the same one based on that sequence, and can be used over and over. But then you start the process of elongation. So in elongation, there's these three steps that always happen. The ribosome slides down how many? 3. It's always 3 because the genetic code is that set of 3 bases. So it always slides down 3 bases. A tRNA, which carries an amino acid. It binds to the mRNA, and you've got to make sure the strands are complementary. That's what's going to determine what fits there. The code on the mRNA matches a specific tRNA, and which amino acid it's carrying is based on this genetic code. So this has been determined. This is consistent. Anytime if you have on the mRNA, you have a UUU, it would be PHE. So that tRNA would be carrying a PHE, which is phenylalanine. If you saw one that was GGG, that would be carrying GLY, which is glycine. So all those abbreviations, if you count, you would see there's 20 different amino acids that are coded for on this chart. And you always just go down by bases of 3. You go down 3 bases, look at that sequence, compare it to the chart, and it tells you which amino acid would fit into that spot. So we're taking the nucleic acid sequence and we're converting it to a protein. We're converting it to an amino acid sequence by using this code. So looking at, once you get 2 amino acids side by side, the ribosome does what? It acts like an enzyme and it forms a peptide bond. So remember, the peptide bond is just what links one amino acid to the next. Where you have a carbon on one amino acid connected to the nitrogen of the next amino acid, all the way consistently down and down. And this just repeats. Every 3 bases represents a new amino acid. So if we look at this, GUG, GGG, CGU, UGU, UUU, ACC, CCU, and then UGA. So in theory, how many amino acids would there be? 10. Okay, so there's 30 bases. That would represent 10 amino acids in the protein. Every time, it's just this elongation. Ribosome slides down 3, new tRNA comes in, peptide bond is formed. Ribosome slides down 3, new tRNA comes in, peptide bond is formed. And every time that happens, the protein chain gets longer by 1. So you're just really just adding a new amino acid on depending on the sequence that you have in that RNA. So go ahead and do that. So at the very bottom, and what I'm going to do, the very bottom is the same sequence. And I actually kind of tried to spread it out just a little bit, and I'll leave this up. So use this to determine the order. So you don't have to draw the ribosome, you don't have to draw the tRNA if you don't want. Just list the order that the amino acids would be linked together in. So we did the first two. So we said that this one is MET, and it's connected to ILE. Methionine connected to isoleucine. So now use that genetic code and determine what are the others. And there's the code. Got them? Okay. All right, so you tell me what's the next one? That stands for valine. GGG? Glycine. CGU? Arginine, that's what it stands for. UGU? That's cysteine. It's the other sulfur-containing amino acid. UUU? Oh, phenylalanine, PHE. ACC? Threonine, THR. CCU? Pro, which is proline. Now, so we get to this last one. And so when you looked on the code, it doesn't have a three-letter abbreviation for an amino acid. It says stop. This is actually it's literally like saying this is the end of the protein. Stop causes the third stage. So the third stage is the end. So initiation is the beginning, and elongation just goes over and over and over, adding an amino acid each time. Termination marks the end of the gene and the end of that protein. So you saw 3 codons. So there's 3 of them in this upper right corner, UAA, UAG, and UGA. And those say stop. That means that they do not code for any amino acid. They do not have a tRNA that will fit. So what happens is a couple try to fit, nothing will fit, and that's the signal to the ribosome to release the new protein, to release the mRNA, and to let it go. And so it's done. So in this, there are 3 base codons that don't code for a tRNA. They call these stop codons. They signal to the ribosome, and it's at the end of the chain. So the ribosome falls off the mRNA and releases this new MET, ILE, VAL, GLY, ARG, CIS, Phi, 3, Pro sequence. That is your new protein. This protein, if it has hydrophilic, hydrophobic areas, it's automatically starting to fold up. If it might form alpha helix, so you might have this helix kind of coming off of the ribosome as it's forming, but it then begins to develop all of that 3D structure. This is that new protein. But once the ribosome releases, it can turn right around and do this again. And so it'll go attached at the AUG. AUG is always attach here site. So that's always where this starts. So it won't attach at the end. It's always going to go from beginning to end in one direction. In fact, you can even have multiple ribosomes on one mRNA strand. So they look like beads on a string. And so they're all just running down, making this protein over and over and over again. So if the cell needs it, think about collagen. Think about hemoglobin. If the cell needs that inside the cell, you make this mRNA strand and it can get used over and over and over to make thousands of the same protein. But every single time, it's the exact same sequence of amino acids are linked together into that protein because you're using the sequence of that mRNA as kind of the recipe or the steps in order to be able to form this protein. So that's how protein synthesis occurs. So that leads us into, and I went through all of these in this as well. I just think it's kind of easier, it's better if you actually write it out. I think you see it a little bit better physically writing it. So the next question is what happens? What happens if the DNA changes?
I got a question. Well, me and Taryn have a question that she doesn't want to ask. But go back to the page you were just on.
There?
And go up. sorry, it's easier to show you. So when do you go from the AUG to the MET? So like, if that makes any sense. To the UAC, like when do you change it?
Here. Okay, so the ribosome attaches at AUG, and as soon as that tRNA lands, then that's the signal that it can slide forward. So it can't move forward until the tRNA binds, that first one that's carrying MET. As soon as that one fits, that's the signal the ribosome slides down. And then the second one can bind. Okay, they can only bind when they're in the ribosome, like coverage, because the ribosome kind of has this shape so that the tRNAs can fit down. So you wouldn't randomly just have tRNAs binding. It only happens because of the shape of the ribosome kind of brings them in and pushes them close together. Remember what enzymes do. So enzymes help to kind of put the two amino acids so that the carbon of one amino acid and the nitrogen of the other amino acid are like right side by side. And that's going to make it really easy for them to get linked together. Because otherwise they'd kind of be floating like this, and you don't know if they would actually ever be able to link together. But that's what the ribosome does. Okay, so it's kind of the combination of the three. The ribosome brings them together. The tRNAs are just the carriers. And then the mRNA is what determines that order that they actually end up being in. Did that help?
Yes.
Okay, all right. Okay, so the last topic is, so what happens if the DNA changes? So if the DNA changes, that is termed a mutation. So mutation always has a terrible term, you know, people are like, mutation, it's a bad thing. Mutation is not necessarily a bad thing, but literally any change in a DNA sequence, that is the definition of a mutation. Just a change. Most mutations are what they call silent. Silent mutations are when your DNA sequence changes and it becomes a permanent thing, but it doesn't end up affecting anything. It just increases variation. So if there is no change in your protein sequence, then clearly it won't affect your life. So on one of your chromosomes, you may get a mutation where like a GC becomes AT. But if it's not in a gene, it's not going to change anything. But it does add to genetic variation. In fact, forensic scientists use this to their advantage because over evolution, over the thousands of years that we have been around, we have had these silent mutations so that average people have small differences in their DNA sequence. And it doesn't occur or affect their ability to survive. But that difference can allow you to take DNA from one sample and compare it to DNA of another sample because the sequences are a little bit different.
Sorry, I didn't think about it, but silent mutations, aren't some viruses capable of don't they change your DNA?
Yeah, so we haven't talked about what causes mutations. That's like 2 slides from now. We'll get there.
Heather.
So it's like, well, it's a good thing. It means you're engaged if you're actually like seeing things that might be coming up. So in this, the thing to remember, very little of the 3 billion nucleotides actually encodes any genetic information. 97.5% of your DNA is just a random sequence. And so having that change in those areas where it's what they call non-coding, non-gene regions, it just adds to variation. So it's estimated that you have 20,000 to 25,000 genes located in your 3 billion nucleotide DNA sequence, but that's really a very small part of the actual sequence. So that's what I'm saying, in that 97%, changes there don't really seem to affect much.
Are the changes there, is that like eye color, hair color?
That would be in a gene. That would be a gene, because genes then would affect how much melanin is made in the hair root. And then that would give you lots of melanin, dark hair, not much melanin, blonde hair. So those would actually be genes. So what we're talking about is part of the sequence that doesn't actually make any protein. But so variations are considered silent. Silent because it doesn't affect your life. It's just going to change your sequence. And those changes that have occurred over thousands of years actually mean that people's DNA is not identical.
How is the junk DNA used to forensic?
Because it's different. So if you get crime scene evidence, so maybe you get sperm or you get saliva or you get hair cell, skin cells underneath somebody's nails, you can extract that DNA and then you can get a suspect's DNA and compare it. So because there is this variation, DNA is much more unique to individuals than was initially thought. So it doesn't mean that it's going, these mutation areas in the non-gene areas just means that we actually have more differences in our DNA than was initially thought or projected. So a silent mutation just means it's a change that doesn't affect any genes. But if it's in a gene, there's a couple of possibilities. If it's in a gene, it still could be silent. If, for example, and I do have to jump back right here. So if, for example, this is the RNA strand CUU, and there is a mutation so that you end up with CUG, is that going to change the amino acid? It's the same. So there's only 20 amino acids and there's 64 possible codon combinations. So 64, some of these codes stand for the same amino acid. So in this case, that would also be silent. Like it wouldn't cause an effect. So it could be that you could have a mutation that codes for the same amino acid. But what happens if you have a code that changes the amino acid? And so the example I give you is with sickle cell. So what do we say happens in sickle cell? So sickle cell is a single gene mutation where you had an amino acid that was polar and was changed to a non-polar amino acid. So that amino acid, remember, would be on the outside. But if it's non-polar, now where's it going to go? It's going to then cause the entire molecule to bend differently and change its shape. Changing its shape changes the ability to bind oxygen, changes how that hemoglobin sits in the red blood cell, creates the sickle banana-looking red blood cells that are very fragile. That's a single change. So that can have very serious effects. So for example, a single mutation, instead of AUU, you have AAU. So that means that just that middle nucleotide was switched, and that would change a non-polar amino acid to a polar amino acid. And that's going to affect the shape. So this can change the shape. And what do you know about proteins? If you change the shape, there's a really good chance you're going to change the function, right? Shape and folding is everything, especially with those globular types of proteins, proteins that act like enzymes. So let's look at a couple other ones. So in here, so see this bottom one, these are all examples of substitution. So here, AUG, that's the same, that'd be methionine. AUC, that's isoleucine. But here, instead of GUG, now we have GUC. Does that do anything? Find GUG. What is that one? So that's valine. But if it switched to GUC, it would still be valine. So that would be an example of a silent mutation. We had a change in the gene, but you still didn't change the amino acid. So keep sliding down. So there's those three. Then GGG doesn't change. That's glycine. CGU is arginine. Now UGU, which codes for cysteine. Now it's UGA. What's that? Yeah, so what is that going to do to your protein? Ribosomes going blah, 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 blah. Hits this, that's the end. So now, there's the end of your protein. So in that one, if that happens, you're going to end up with a protein that's like short. What do you think that's going to do? Think it'll affect its shape? Yeah. So it'd be like cutting the end of your key off. Is your key going to work? No. So you took the whole end piece off. So that could end up affecting function. So it could cause, they call that like premature termination. So that could cause a protein that's really short. So what if that one didn't happen, but then, so we kept going on normal. UUU is phenylalanine. The ACC, that one I can't find, is threonine. And then we have CGU, or CCU, sorry. So CCU is normal, it's proline. But this one, UGA, normally means stop. But here, we've changed the U to an A. So what's AGA? Is that a stop? Yeah, it's another arginine. So what is that going to do? That's going to tell the ribosome to keep going. So if there is anything else on this strand, it's going to end up making a protein. So instead of having just those 9 amino acids and then you hit stop, now you might have 13, 14, 15. You could end up with a really long protein chain, and that would be like trying to put trying to stick your house key in a little master lock. So having this really big key, there's no way it's going to fit. And so that could end up leading to a long protein. So elongated proteins, shortened, change in shape, because you're switching like polar amino acids for non-polar amino acids. All of those things, great chance that you're going to change the function. Now, some of these changes that evolved over time ended up giving us more diversity, ended up giving us more characteristics, more traits. So things like you may have a gene where you make melanin. So that melanin protects you from ultraviolet light. So if you're out in the sun a lot, you want to have more melanin and you get better UV light protection. It reduces your chance of getting skin cancer. And so you saw that people that were outside more, those genes were active more and they ended up with darker skin color. What about what causes these? So like I said, sometimes it just happens. Spontaneous, random, just random mutations. So when the cell is going through normal division, there's an error. You have to take 3 billion base pairs, ATGC, and copy them in order for a cell to divide. If you had to copy 3 billion letters, do you think you would ever make a mistake? 3 billion. Yeah. Yeah. And you actually you copy this, enzymes that do this, copy it at the rate of about 100,000 nucleotides a second. Like it just like zips down the DNA and like assembles a complementary strand based on the old strands. It's really fast, how quick it occurs. So yes, there's spontaneous errors that happen. In fact, most of them get fixed because if normally you were GGCT and so the strands were like this, so we would have CCGA. That would be like the normal DNA. If there was an error when you did this replication, and instead of GGCT, we got CGGA. 2 G's, what are they going to do? Are they going to hydrogen bond? No. And in fact, what they do is they won't fit together like everything else, and it forms like a bulge. So the because the GG can't fit together like GC does, it'll cause the helix to have this little bump in it. And so that actually helps to improve repair. So there's enzymes that are always feeling the DNA, looking for mistakes and fixing them. So most of your spontaneous errors get fixed, but sometimes they don't. Sometimes the cell goes on and continues division, and then you end up with a mutation that stays. So other things that cause, there's environmental agents. So we can have spontaneous mistakes, just because the cell's really busy and it does make mistakes. Environmental or ultraviolet light. UV light is a good example of a mutagen because it's energy, and that energy can cause damage to DNA. And that damage, if you go out and you get a whole lot of ultraviolet light exposure, what do you end up with? A sunburn. And then you're like walking around and then that all peels off. So you actually like kill those top layers and all that peels off. But ultraviolet light can cause damage to a certain type of cell in the skin called a melanocyte. Melanocytes are what make pigment. And so if this cell gets damaged, it may actually lose control over its cell cycle. So one part of the DNA's job is to control when the cell divides, to make sure that cells divide appropriately when there's room, when there's nutrients. And so mutations can cause abnormal cell growth. And abnormal cell growth leads to cancer, tumors, masses, cancer. So ultraviolet light is one of them. Other ones, like you were saying, viruses. So viral infections, and not typically like your common cold type of viruses, but viruses that actually infect internal cells versus like surface cells. These types of viruses infect a cell, and they actually insert their genetic information, their DNA, into your DNA. And it tells your cell, make lots of this virus. So it literally takes the viral DNA and turns it into a gene that then gets turned on constantly. And what it does then is it tells your cell, make lots of virus particles, and when the cell starts to fall apart, all those virus particles get released. So that's really like the end of this chapter. We're really not going to talk about it. It's really kind of like fascinating how viruses can work. But when they insert, now they might insert in a gene. And then when they pull the DNA piece out, it can cause changes. So viral infections can lead to DNA mutations because virus particles are present within the nucleus.
Is that why strep throat can cause issues with your heart?
That's really, with strep, what happens, that's an immune response. So that's because the strep bacteria get stimulated by your immune cells, but the surface of the strep bacteria are a lot like the surface of your heart valves, and your immune cells really can't tell them apart, and they attack both. So that's kind of an autoimmune response with rheumatic fever. But with viruses, like examples of this, where a viral infection actually ends up causing like another cell to die. I think of like type 1 diabetes, like where they have cell gets infected, either immune system attacks and destroys it, or that viral infection causes so much damage to those pancreatic cells that they quit.
What pancreas started, when I was in 6th grade, I got a viral infection, and they couldn't tell me what it was.
Mm-hmm.
And winter came along, I had colder to carry it now.
Mm-hmm.
And I never did blood work to find out that it was actually a virus, but they were like, yeah, it's probably a virus.
Mm-hmm. Yeah, so it just depends on the type of virus and where it actually inserts, and then if it actually causes a mutation that could lead to like a long-term response, like long-term issues with them. Last one are chemicals. So chemicals that have the ability to cause changes in the DNA sequence are called mutagens. So think about these 3 different types. What is their goal? What do sides do? Insecticides, pesticides, herbicides, they kill things, okay? So the goal with most of these types of chemicals that they develop is they want to find something that the bug or the plant does that humans don't, so that they can actually then spray it and it can be around you and not end up having a negative effect. But what is one that they have just started looking at that has been used by millions of people? Roundup. How many people have sprayed Roundup? Mm-hmm. So Roundup, like I hate a weed. I'm just going to say right now. Okay? That, when it was introduced, it is the most safe of all of the herbicides. You could take a bath in it and it wouldn't be a problem. And now, 20 years after they've been spraying it everywhere, all over your food, okay? So they like spray it as a way to like defoliate. So they kill the plant so that then they can harvest. So after doing all that, now they're starting to show, well, there is some signs that it could actually cause mutation. Great. Okay? Not as much of a problem. I mean, it can cause an issue, like for you, like if you have a mutation in a body cell, that means that one cell may begin to function abnormally. So that's just a single cell. Yes, that could lead to like melanoma, or that could lead to like recurrent issues. But this is really an individual cell. So a single cell gets affected. So that could be a skin cell, or that could be a pancreatic cell. That could be individual cells that are affected by a mutagen. But if you have a mutation in a germ cell, and by germ cell I don't mean like bacteria. Germ cells are your sex cells. Germ cells are eggs and sperm, or cells that develop into eggs and sperm. These, if you have a mutation in an egg, and it gets fertilized, what is that going to mean? That means that now the offspring are going to have that genetic mutation will be part of their genes. That is going to be part of their DNA. It's not going to get repaired because it actually happens at the germ cell level, and that leads to these genetic diseases. So when you go get an x-ray, what do they put over your pelvis? Lead. Okay? Because they are trying to protect those germ cells, more so than somatic cells. So somatic cells, that cell might die or that cell might grow abnormally, it might develop into a mass. But with germ cells, that means that you could have offspring that are going to have genetic diseases. And like there's like the list. And this is by no means, it says there's like 4,000 plus genetic diseases. And so sickle cell's one of them. So somewhere along the line, there was a mutation that was introduced to a germ cell that affected the hemoglobin gene and led to somebody that ended up with abnormal hemoglobin. List of them. You may know some people that have this: cystic fibrosis, Down syndrome, muscular dystrophy, Huntington's disease. In fact, BRCA, the BRCA genes, those are the breast cancer related genes. So having these genes, it's really having kind of like a mutated normal gene. Having those genes increase your risk of developing breast cancer. So they find in families that have these genes that women have a much higher incidence of breast cancer. So if you find that like family members develop breast cancer, they may want to test you for the presence of those genes because that ups your risk factor. My mother was 47 when she was diagnosed with breast cancer, ended up having a mastectomy. She's now 78, and so has had no problems or issues since. But because of that, she was tested, but she was negative for it. My sister got tested. She's negative for it. So in that case, with my mother, it may have been an environmental exposure. It may have been something else that caused that abnormal mutation in one of those breast cancer or one of the breast cells that caused that abnormal growth to occur. So in that one, there wasn't a link, but they do find that in families. So with these, there can be family links to them. There's sickle cell. Okay? So those are just some of the genetic mutations that can occur. So that's where you have random, just random spontaneous, and that could have been the reason that she developed breast cancer, just random spontaneous mutations. Then you can have things like ultraviolet light, viruses, and then chemicals, chemical exposure. So Roundup's an interesting one. Another interesting one that you probably don't know about because you're all too young. But back in the 40s, M&Ms always had red color. So they always had red M&Ms. And then they found out that that red dye was a mutagen. So all these kids, and so then it was like, so they like took all red M&Ms. And when I was a kid, there were no red M&Ms. They didn't have red M&Ms for about 30 years because it had such a negative stigma that that red dye actually acted as a mutagen and could cause damage to the GI tract. So they reintroduced it around 2000 where the red M&Ms now are put back in. But there was a good 30-year span when you never saw a red M&M. So it was just sort of an interesting fact. But that's just an example of a chemical additive in foods.
What about, as far as beauty, what about going to get your nails done and the gel nails?
And you have your fingers.
Yeah.
So all of those ones, it's pretty short-term exposure. So the amount of time that you're exposed to, man, I'd be more worried about all the stuff you're smelling when you're in there. Because you feel like you've been huffing by the time you leave the nail salon, just from all of the solvents and everything that they use. All right, so that finishes up. So go ahead. Like I said, I left pre-lecture 11 and 12 open. If you hadn't done them, make sure that you go back and check. You can go ahead and do the post-lecture homework for 11, do the dynamic study module for 11, and then